And ARDNA is the DNA binding subunit of the BAF chromatin remodeling complex, which is part of the SWI/SNF family. And these complexes function in the regulation of various processes within the cell, including gene expression, by either directly remodeling chromatin by sliding nucleosomes around, or via the recruitment of other factors to regions of the genome. And as I mentioned, ARDNA is specific to the canonical form of the BAF complex and acts as the DNA binding subunit. And as you might imagine, ARDNA loss in cancer can result in the dysregulation of many processes, including epigenetics, where we usually see a general decrease in chromatin accessibility. There's other phenotypes that include gene expression dysregulation, which is often characterized by transcriptome rewiring and transcriptional dynamic defects. Our lab has also reported on the consequences of ARDNA loss on genome instability, where we've seen increases in things like DNA replication stress and DNA damage, which are partly explained by an aberrant accumulation of R loops. And it's also worth mentioning that ARDNA has a paralog called ARD1B, which can partially rescue some of ARDNA's function within the BAF complex, but obviously not entirely, because we see all of these things happening. And although the study of ARDNA mutations in specific cellular contexts has allowed to identify vulnerabilities of ARDNA deficient cells, and I've tried to illustrate some of these in these red boxes around, there really hasn't been any major breakthroughs in the clinic for the treatment of these cancers. And ultimately, there remains a need to identify new ways of selectively killing these ARDNA deficient cancer cells. So that's where my project comes in. And so my goal is to address this issue by identifying synthetic lethal partners of ARDNA as new therapeutic targets using genome-wide CRISPR screening. And so synthetic lethality is this concept where a combination of genetic deficiencies will result in a lethal phenotype, whereas a single deficiency will not. And so in other words, we know that cancer cells select for the loss of ARDNA. And if we're able to identify synthetic lethal partners, for example, gene B in the slide, that we could target using a drug, this would allow us to selectively kill these ARDNA deficient cancer cells. So to do this at the genome-wide scale, I've employed the Toronto Knockout version 3 library, which is a CRISPR library that targets 18,000 genes in human cells. I've packaged this library into lentiviral particles, which I've then used to infect ARDNA wild-type or knockout cells at a ratio so that only one guide RNA should be incorporated per cell. And so the infected cells can then be selected for with pyrimycin, and we collect an initial time point for the experiment to assess the representation of our library at the start of the experiment. We can then outgrow the cells for about 14 days to allow time for fitness defects associated with these guide RNAs to develop. And then using amplicon sequencing and the Bagel2 algorithm for analysis, we can compare the guide RNA representation at the start of the experiment versus at the end of the 14 days in both our ARDNA wild type and knockout populations. So you can imagine that if a guide RNA causes a fitness defect, we would expect this guide RNA to drop out over time as the cells die. And so as such, if we see a guide RNA that drops out in both the ARDNA wild type and knockout cells, we could infer that this is potentially an essential gene for these cells. Whereas the guide RNAs that drop out specifically in the ARDNA knockout cells are potentially synthetic lethal partners of ARDNA, and those are the hits that we are interested in. So I've performed this CRISPR screen approach in biological duplicates in an isogenic cell line model of ovarian closed cell carcinoma. And I've looked at the precision recall curve to first evaluate the performance of our screens. And so overall, we found that our experiments captured over 99% of the guide RNAs in the library at the initial time points, which gave us confidence that we infected our cells with the appropriate representation of our library. And the high precision to recall ratio plotted here that you see suggests that our algorithm was able to identify a relatively low false positive rate and a low false negative rate based on training data that it uses to give out Bayes factor score, which are an indication of fitness. When we overlap the hits between the two replicates of our screen, we find, and so I'm showing here fitness genes that are associated to ARDNA deficient cells specifically, we find that there is a significant overlap of 103 genes between our replicates that we've dubbed our consensus data set of AirDNA synthetic lethal partners. One of which is KEAP1, and I'll touch upon that very soon. Unfortunately, if we do a Go analysis on this consensus data set, it doesn't really return anything significant in terms of enrichment because of the small sample size. But if we look at the entirety of the genes that are important in either replicate for AirDNA deficient cells, we find that gene expression related processes, mitosis, splicing, mitochondrial processes are all enriched, which agrees with the literature around AirDNA loss in cancer. And it's also worth mentioning that we did recover some known synthetic lethal partners of AirDNA among our lists. If we average the base factor scores or the fitness scores that we've obtained for our consensus data set from each replicate, we find that the top 3 hits from our screen are PNPO, CTDSP1, and KEAP1. And today I'll talk about KEAP1 because it's one of the targets that I decided to validate. So KEAP1 is part of an E3 ubiquitin ligase complex, along with CULT3 and RBX1. And KEAP1 regulates various processes within the cell, including proteostasis, cell cycle, and more. But one of its most studied functions is its role in repressing the transcription factor NRF2 by targeting it for proteasomal degradation under normal conditions. Under conditions of oxidative stress, KEAP1 C-terminus can get oxidized, which will trigger the release of this inhibition of NRF2, which can then translocate to the nucleus to activate the transcription of hundreds of genes. So the first thing I set out to answer from our screen in terms of validation was if AirDNA knockout cells are actually sensitive to KEAP1 depletion. So the first thing I did was to knock down KEAP1 with sRNAs in our isogenic RMG1 cell line pair that we did the screen in. And so as you can see, knockdown of KEAP1 with 2 different sRNAs resulted in less protein and an induction of NRF2 levels, which was expected. And you can also see here that the AirDNA knockout cells in red were sensitive to this inhibition compared to the wild type. To complement this data, I also generated an AirDNA knockout clone in RP1 cells using CRISPR so to have a second isogenic cell line pair to test on. And again, we observed very similar phenotypes in the cell line where AirDNA knockout cells were sensitive to the knockdown of KEAP1, but not for the wild type cells. In addition to the RNAi data, I also used small molecule inhibitors of KEAP1, one of which was called AI1. So as you can see from this InQSight data, AirDNA knockout cell growth was impaired by AI1 inhibition. So on the left, I'm showing data from our RMG1 pair, and on the right from the RP1 isogenic pair. So the knockout cells are shown here in red or orange. And as you can see, AI1 inhibition resulted in more significant growth defects in the AirDNA knockout cells. So overall, these findings supported the validity of the hits from our screen and sort of confirmed that KEAP1 was in fact a synthetic lethal partner of AirDNA. And the next thing I wanted to do was to look at what happens to AirDNA knockout cells when we inhibit KEAP1. And being a genome instability lab, looking at replication stress phenotypes was a good place to start. And so I first looked at genome instability markers in AirDNA wild type and knockout cells in the presence or absence of this KEAP1 inhibitor AI1. And what I found is that overall, the AirDNA deficient cells do have more replication stress measured by phospho-RPA serine 33 foci formation, more DNA damage compared to the wild type measured by gamma H2AX formation, and more micronuclei under normal conditions. And this is all in agreement with previous data that we had generated and published in the lab. We found that if we treat the cells with AI1, this KEAP1 inhibition results in an exacerbation of these genome instability phenotypes, and this is specific to the AirDNA knockout cells for all cases. And so it remains unclear what's at the root of the mechanism between the KEAP1 and AirDNA deficiency that we've identified, but we're working on a few hypotheses to better understand the mechanism, and it appears that this exacerbation of genome instability in AirDNA deficient cells following KEAP1 inhibition could perhaps be utilized therapeutically. And to sort of further test what mechanism could be at play here, we wanted to look at the sensitivity of ARID1A deficient cells to AI1 and see if we could rescue it by knocking down NRF2 as a way to explain perhaps how this synthetic lethality was working. So as I told you earlier, NRF2 is the downstream target of KEAP1. So KEAP1 inhibits NRF2 under normal conditions. And if we use the KEAP1 inhibitor AI1, we would expect an induction of NRF2 transcription in the cell. So the data I'm showing you here is the confluency of cells after 60 hours of treatment with AI1 relative to SI control conditions. So the RNG wild-type cells are shown on the left 3 columns, and the ARID1A wild-type cells are shown, the ARID1A knockout cells, sorry, are shown on the right 3 bar graphs here. We have the SI control, SI KEAP1, SI NRF2 for both. And what you can see here is that the RNG wild-type cells don't present any significant defects if we knock down SI KEAP1 or NRF2. However, in the wild-type cells, we see a fitness defect if we knock down KEAP1, which confirms our findings from earlier. And we actually see an increase in survival if we knock down NRF2. If we treat the cells with AI1, we again don't find any significant phenotypes in the ARID1A wild-type cells, excuse me. However, we do see a sensitivity for the ARID1A knockout cells under control conditions, which is what I've showed you in the past. If we knock down KEAP1 in these cells, we see more significant fitness defects, which was expected. And interestingly, if we knock down NRF2, this rescues the survival of these ARID1A deficient cells, suggesting that perhaps the dysregulation of NRF2 might be the cause of the synthetic lethality in these ARID1A deficient cells. And this is also true at higher concentrations. So we're testing this hypothesis at the moment with alternative approaches, but this so far is sort of our main idea regarding the mechanism. It's that NRF2 dysregulation is somehow toxic in these cells. And so finally, before I end, I just want to present one last piece of data that we obtained with some collaborators in the Huntsman lab here at BC Cancer. So one of the questions we wanted to address was if our findings were restricted to our cell line models or if there was a broader application to this genetic dependency we were studying. So the Huntsman lab has developed this protocol to culture ARID1A knockout organoids derived from patient samples. So what they do is they obtain patient-derived endometrium progenitor cells, which they can then transduce with lentivirus that contains a CRISPR guide RNA targeting ARID1A to generate these ARID1A knockout organoids, or a non-targeting control, which shouldn't affect ARID1A status. They can move these cells into organoid culture where they grow into these nice 3D spheres, as you can see here on the left, and they contract these transduced patient-derived organoids because they glow red using a TD tomato reporter. So you'll see arrows on these images that highlight some of these TD tomato transduced organoids, and they can treat these organoids after that with KEAP1 inhibitors to assess survival and morphology. And that's exactly what Faru did in the Huntsman lab. So as you can see from these images and the quantification below, Faru has treated these organoids with increasing concentrations of the AI1 and confirmed our findings from cell lines where ARID1A knockout PDOs present significant survival defects compared to the wild type or the non-targeting control. And I've blown up this 75 micromolar concentration for you to see a little bit better. And you can follow with the red arrows these TD tomato organoids that are much smaller and much darker in the ARID1A CRISPR fraction compared to the non-targeting control. And this suggests that they experience growth defects, and the dark color suggests that they're perhaps undergoing necrosis. So overall, this suggested that our KEAP1 ARID1A dependency was not restricted to our cell line models, and maybe it has more significant clinical potential that we are exploring at the moment. So I'll conclude here. Overall, I've uncovered a new genetic dependency between ARID1A and KEAP1. I've also showed that KEAP1 inhibition exacerbates genome instability phenotypes in ARID1A knockout cells specifically. In terms of mechanism, we believe that NRF2 dysregulation may contribute to the sensitivity of AirDNA knockout cells, as it was seen with our NRF2 knockdown rescue data, but the exact mechanism remains unknown. It's possible that it's because of transcriptional dysregulation through NRF2, and so imbalanced levels of NRF2 would be associated with things like changes in metabolism, also things like productive stress. So we're looking into these possibilities at the moment. There's also possibilities that other functions of KEAP1 could influence these phenotypes. So there's evidence that KEAP1 might control TOP2A levels, and this could lead to topological stress in these cells. We've reported in the past that AirDNA knockout cells have TOP2A localization defects, so that's another possibility. It's also possible that it's through the dysregulation of proteostasis, as KEAP1 regulates degradation of various proteins and has roles in autophagy. So the mechanism is still very much in the works, but we are working to try to get a better understanding of what is happening with this dependency. So with that, I'd like to thank everyone involved in our lab, my supervisor, my thesis committee, and our funding agencies. And I'm happy to take any questions. Thank you.
All right, thank you for that great talk, Louis. So if anyone has any questions, feel free to raise your hand and I will be unmuting you. Go ahead, Matt.
That was a really nice talk. Thanks. That's really cool.
Thanks.
Thinking about the NRF2, do you guys think it would be useful to do like a transcriptomic analysis to see if in the ARID1A knockouts you're actually getting transcriptional changes that kind of correspond to NRF2 dysregulation?
Totally. Something that I didn't mention is we actually collaborated with a lab at UBC to do RNA-seq analysis of AirDNA wild type versus knockout cells. So someone in the Hearst lab looked at a publication from 2019 that was using the same cell line model as we were for our AirDNA knockouts in ovarian clear cell carcinoma. And we reanalyzed this data and found that actually NRF2 target genes are both dysregulated in the, or both upregulated and downregulated significantly in these AirDNA deficient cells. So that suggested to us that there's probably a role for NRF2 in this whole story. Something that I was thinking that could be interesting to do was to look at these cells treated or untreated with AI1 and look at sort of by mass spec, how the proteome changes from that. It's not something we've gotten to do yet, and it's a bit past the scope of our study at the moment because I'm trying to wrap up, but it's something that would be super interesting to look at.
Yeah, that's cool. Thank you.
Yeah, thanks.
I see we have another question from Dr. Affar. Go ahead.
Yeah, well, thank you, Louis-Alexandre, for the great presentation and the nice work. I was wondering about the, did you see, I don't know if it has been done or if you're planning to do that, to see in cancer how ARID1A and KEAP1, for example, are co-expressed or mutated. It might be even in a mutually exclusive manner. Do you have some plan for these experiments?
So I did look at that. Unfortunately, I don't have an extra slide for it to show you at the moment, but it doesn't seem there's a general inverse correlation between ARID1A levels and KEAP1 levels across all cancers. It's quite mild in terms of relationship or correlation. I've also subsetted cancer subtypes by ARID1A mutation status and looked at KEAP1 expression versus in ARID1A mutated versus wild type cancer subtypes. And I found that some cancers do upregulate KEAP1 when ARID1A is mutated. But the inverse is also true for different subtypes. So it's not a clear-cut relationship, but it's probably applicable to different cancer subtypes. For example, colorectal is one of the differentially, I guess, expressed KEAP1 and ARID1A levels that I remember of.
Thank you.
Yeah, thank you.
All right, so in an effort to keep us moving along, I ask you save any questions or message Louis directly. And now I just want to get our next presenter, Dr. Affar, to share his screen.
Do you see my screen?
Okay.
Okay.
Yep, now it's good. Perfect. Okay, so I'm going to let Dr. Affar take it away with his talk titled Liquid Phase Separation of the Mammalian Nuclear Proteasome Links Amino Acid Supply to Apoptosis.
Well, thank you. Thank you, Patty, and thank you to the other colleagues helping you to organize this nice event that we have once a month by the CSMB. It's really great to have this kind of seminars for interactions. I'm very happy to present today what we have been doing for the last few years in the lab. I will be talking about some already published results and some new results that we have on the mammalian proteasome, on the regulation of the mammalian proteasome by liquid phase separation in response to changes in amino acid supply. So just briefly, I will introduce ubiquitination. This is a post-transcriptional modification that involves so this is a post-transcriptional modification that involves three enzymes, E1s, E2s, and E3s. E3s are responsible for the specificity of the ubiquitination reaction, and these are the enzymes that we recognize the substrate to ensure this ubiquitination reaction. So we have a lot of ubiquitin ligases in the human genome. They are involved in many cellular processes. We have thousands of cellular substrates. So it's not surprising that ubiquitination will be not only involved in many cellular processes, but also defects in this post-transcriptional modification will be associated with human disease, and notably cancer and neurodegeneration disease. So again, just briefly in introducing ubiquitination. So ubiquitination can come in different flavors. We can have monoubiquitination of substrates. So this is basically one molecule of ubiquitin that can be attached to substrate. We can see that in endocytosis, transcription, DNA repair mechanisms, but ubiquitin itself has 7 lysine residue in addition to the N-terminus of the ubiquitin that can be used for chain extension, for polyubiquitination reaction. And we have, for example, ubiquitination through the lysine 48 of ubiquitin. So this ubiquitination is mostly associated with protein degradation. We can see that, for example, during cell cycle progression, destruction of cell cycle regulators. And in contrast, we can have another polyubiquitination, for example, here through lysine 63 of ubiquitin, which is not generally involved in protein degradation. We can see that, for example, in protein activation, in DNA repair and kinase signaling. So we have the ubiquitination that we bring to degradation by the proteasome, but ubiquitination can also have some proteasome-independent events which are not associated to proteolysis. So the proteasome itself, it's a fantastic, actually, protease that is responsible for the degradation of unwanted, I will say, cellular proteins, and we have the catalytic particle of the proteasome, we have the regulatory particle of the proteasome, which will be responsible for unfolding protein substrates, removing the ubiquitin from these substrates and channeling these proteins for the catalytic chamber, the 20S, for degradation to release peptides that they can be used to generate amino acids, and these peptides can be also part of the immune response. So we have another regulatory particle. I'm mentioning that, and we have some additional ones. I'm mentioning it because it will be relevant for the rest of the talk. And this is PSME3 11S, which is also involved in regulating the function and the catalytic activity of the proteasome. So one of my students made this observation that when we take the cells and we deprive them from nutrients, we see that the proteasome can form these very nice foci in the nucleus. So we have PSMD14 staining, which is a component of the regulatory particle of the proteasome. So we have been intrigued by this observation, and actually I want just to define that the nutrient starvation, the nutrient deprivation that I will be using throughout my talk. It will be a buffer, basically. It's an HBSS buffer which contains glucose but doesn't contain amino acids. So most of the work that I will display today has been done by Maxime Uriart, who recently finished his PhD, and he's looking for a postdoctoral position, and he did really a great work to characterize this proteasome foci. First of all, he wanted to know whether we have actually the proteasome in the full proteasome in this foci. So he used a lot of antibodies for endogenous proteins, and in each time he can see the co-localization of a component of the catalytic particle with the component of the regulatory particle. For example, PSMB1, PSMD11, PSMD4 with PSMB7, PSMB2, D11, B4 with D11. So there is really a nice co-localization with all these components of the proteasome in the nucleus upon nutrient deprivation. He can also see that PSME3, this another proteasome regulatory particle, can be also co-localized in this proteasome foci. He can do that also with PSMB4 GFP, so he generated the fusion protein of PSMB4. He generated additional fusion proteins for the catalytic particle or the regulatory particle, and in each case he can see that we have a very nice foci in the nucleus upon nutrient deprivation. So now he wanted to know whether the proteasome integrity is required for the formation of this proteasome foci. So we, for example, deplete a component of the proteasome, for example, here D11 and D4, or PSMB5 and PSMB7. And in each case, you can see that the cells lose the ability to form this proteasome foci. So next, he wanted to know whether this is a general phenomenon that can be observed in different cell types. So he took many mammalian cell types. It can be primary cells, and when he does his nutrient deprivation, he could see this very nice foci forming in the nucleus. He can also see that in immortalized cells. Again, with nutrient deprivation, we can see nicely the foci. And in cancer cells, many cancer cells, we can also see this localization of the proteasome in distinct nuclear foci in the nucleus. He can see that also in mouse embryonic stem cells, and he can see this foci also in cells before and after differentiation here from pre-adipocyte to differentiated adipocytes. You can see nicely that the proteasome form foci after nutrient deprivation. So this seems to be a general phenomenon, general response of at least mammalian cells to nutrient deprivation. So he wanted to characterize whether these proteasome foci correspond to other described foci in the nucleus. So he characterized by costanins for example, PML bodies, nucleoli, speckles, DNADMH foci, and in each case, he can see that these are distinct foci of the proteasome that are independent of other known foci. And he can see nicely here with PSMB GFP, again, a component of the catalytic component of the proteasome, that these foci can form in chromatin-poor region of the nucleus. And with electronic microscopy, we can see that these nice foci are membrane-less organelles that form after nutrient deprivation. At this stage, we decided to give them a name, and we named them SIPAM for starvation-induced proteasome assemblies in the nucleus. And during the same time when we were doing this work, a Nature paper has been published to report that upon osmotic stress, proteasome form foci in the nucleus. And it seems that they are more or less the same foci, but I think more effort has to be put to understand what is the exact composition of these foci and what is the difference between these foci that happen upon nutrient stress and foci that happen after osmotic stress. You guys know about this liquid phase separation and this idea that proteins or macromolecules, they will segregate in cells, in the cytoplasm and in the nucleus to form distinct entities to catalyze specific or to inhibit specific functions. So the way to think, or at least my way of thinking about this liquid phase separation and why it can be important for cellular processes, I will give you an example. For example, with water, solid water, ice. When we put ice in water, we can say that we have here a solid liquid phase separation. We have two phases, one solid, one liquid, and basically there is no mixing. So the ice has to melt to have that eventual mixing. In respect to the liquid phase separation, we can think about oil and water. So oil form droplets into water, and you can think about it in the way that, you know, in this sense, since we have liquid phases, we can think about mixing them. We can think even about exchange of components between the two phases. So this is my way of thinking about this phase separation. And we can apply that now to macromolecules or to the proteasome. And this is with the idea to say that we will segregate proteins. We form this phase separation, these droplets, these membrane-less organelles, and that can be basically involved in, for example, activating a cellular process, inhibiting a cellular process, or changing the localization of proteins to, again, catalyze or inhibit cellular functions. So we wanted to know whether this separation of the proteasome in the nucleus under conditions of nutrient deprivation is a liquid phase separation. So first, we try to characterize these foci. For example, here we can count them as a function of time, and you can see here in this cell type that these foci can form after a few hours, and they reach maximum after 6 to 10 hours after nutrient deprivation. We can also measure the air intensity of this foci, and we can count the number of foci per cells. We have in general around, in average, 40 to 50 foci per cell, and you can notice that upon time, the number of foci reach a maximum and decrease after that. And this is because we have fusion events, which is one of the characteristics of the liquid phase separation. And as you can see nicely here, these two foci have fusion to form these foci, and these two foci have fusions to form these foci. So we have fusions of these foci, and you can, as you can see here, this is concomitant with an increase of the intensity of these foci. So we wanted to know more about their dynamics and whether we can qualify them as liquid phase separation. We went to understand or to investigate more about their dynamics. So here, for example, when we take this phase separation, this foci that they form after treating cells with HBSS, and we put them in normal media, we can see that these foci dissipate. They dissipate very quickly after 40 minutes or 60 minutes. We have almost a total dissipation of this foci. Of course, the intensity of foci will decrease as well. And we took actually advantage of this assay to know or to determine whether the VCP, the P97 chaperone, whether this chaperone is required for the dissipation of this foci, and the response is no, because we used different inhibitors at different concentrations, and we cannot prevent this foci dissipation with these inhibitors. So we used after that, this is with the idea of continuing our characterization of this liquid phase separation. We used this compound called exendiol, which interfere with hydrophobic interactions. And as you can see here, with higher concentrations of exendiol, we can see nicely that this foci dissipates, supporting the notion that these foci are a consequence of a liquid phase separation. And we can do another assay by taking basically these foci and incubating, taking the cells and incubating them in hypotonic buffer, and we can see that this foci nicely dissipates following this treatment in a hypotonic buffer. And I have to point out that at this point we don't have any impact on the integrity of the cells. In fact, when we add when we add a medium, when we put an isotonic buffer, we can see that these foci reform again, and in their, actually they form in their original location. So these foci are very dynamic, and they have all the characteristics of liquid phase separation process. We also try to investigate more their dynamics by bleaching them with laser. And as you can see here, for this foci, when we bleach the foci with the laser, we can see a very quick recovery within 10 seconds. We can see a nice recovery in the original foci. However, for example, 53 BP1, which are DNA damaged foci, when we hit them with the laser, we don't see a recovery during this time frame. So these proteasome foci are highly dynamic, and they, with all means, I think we can say that they are the result of a phase separation and liquid phase separation. And we wanted to understand more how these foci form and what is the consequence and what is the impact on the cells following the formation of these foci. So one of the things that we did is to try to understand the role of ubiquitination in this process. And one of the first things that actually we did is to determine whether the proteasome is active in this foci. And we did it two ways. We selected nuclear proteins, for example, here CFOS and CJUN and P53, and we took cells that they form nicely the foci after 6 to 8 hours of treatment, and by adding MG132, we can see that we have an accumulation of CFOS, CJUN, and P53, suggesting that the proteasome is still active in this foci. We use also this probe, this proteasome activity probe, and you can see nicely the co-localization of this activity probe with the proteasome foci. So at this point, knowing that we don't have definitive proof that the proteasome is active in this foci, but at least with this experiment on checking the stability of proteins and with the probe, we can say that we have activity in these proteasomes. But we can't say that these proteasomes in the condensates at this point are more active, for example, than the soluble proteasomes in the nucleus. So we wanted to understand more about the ubiquitination and how the ubiquitination is regulating this process. So one of the first things we did is to see whether we have ubiquitin in the foci, and in fact we do. This is ubiquitin K48 that we can see nicely in the foci, and when we treat with this E1 inhibitor TAC243, we can completely dissipate this proteasome foci. In addition, when we take cells with foci and we put either the proteasome inhibitor or a DOB, a general DOB inhibitor, we can see that with the inhibition of DOBs, we prevent the dissipation of this foci. So this strongly suggests that ubiquitination is involved in their formation and that de-ubiquitination is required for their dissipation. So we wanted to know whether we can identify factors that can be involved in this liquid phase separation of the proteasome. We considered several shattering factors. So these are proteins that will bring ubiquitinated substrate to the proteasome, and we did RNAi for several of them, and one of them, actually RAD23B, when we knock out or knock down RAD23B, we can see that we have a very nice or dramatic, I will say actually, decrease of foci, of proteasome foci formation. And in fact, RAD23B co-localize with the proteasome, nicely co-localize in this foci. So we wanted to determine, because this RAD23B was a very good candidate for a protein that will promote this phase separation. It has disorganized regions. It has also organized regions. So one of the things that we did is to remove the domains and determine the ability of RAD23B to promote this phase separation. And as you can see here, by basically removing the critical domains, we didn't touch yet the non-organized regions. We can see that we completely abolish the ability of the cells to form this liquid phase separation of the proteasome. And in fact, we purified RAD23B, and this we can induce droplets in vitro when we add the crowding agent, as you can see here, the solution become turbid. We can see the droplets, and we did the phase separation diagrams by changing the concentration of the protein and the crowding agent. And we can also see that when we delete the domains, as we can see mostly in the cells, that when we remove the critical domains of RAD23B, we can abolish the ability of the RAD23B to form these droplets. So just a conclusion at this point. So the proteasome undergoes liquid phase separation after amino acid deprivation. These organelles that we call cypan are highly dynamic and quickly dissolve following amino acid replenishment, and that ubiquitination and de-ubiquitination are required for cypan formation and dissipation, and that RAD23B drives this phase separation. I think we don't have that much time, so I will perhaps go quite quickly on some of my slides. My apologies for that. So to make a long story short, to determine how, and we don't know yet how the depletion of amino acids induce this liquid phase separation, but we can manipulate amino acids by blocking protein synthesis, by blocking autophagy, and by adding or moving amino acids. And what we found is essentially non-amino acids, non-essential amino acids are the major determinant in this phase separation. In fact, we took the amino acids one by one, and we did our starvation experiment, and mainly the non-essential amino acids can prevent this liquid phase separation of the proteasome. We can see that for the formation of the foci and also for the dissipation of the foci. So to determine the significance, what does it mean to have this separation of the proteasome, this liquid phase separation? We don't know what's the significance of that at this point, but all I can tell you that it seems that it is an important event, and if we interfere with two proteins, RAD23B and PSME3, and by doing RNAi for these proteins under conditions of nutrient deprivation, we promote the survival of the cells, meaning that the liquid phase separation of the proteasome under conditions of nutrient deprivation will be promoting cell death, as you can see here with this phase contrast, these MTT assays, and this assay for PARP cleavage. We don't have that much time left. I will go just quickly to the perhaps to this. I think it's a nice result that we had recently, and we are characterizing more in respect to identifying and looking at this liquid phase separation of the proteasome in vivo, in tissues. And as you can see here, this is a tumor. This is a tumor in mouse, and we can see here that in regions of the tumor that they have more apoptosis, we can see nice phase separation, or at least condensates of the proteasome in cells. So we know that these organelles can also happen in vivo, and we are characterizing some other context where we can see these organelles and interfere with them. And just maybe to conclude my presentation with this, So why we want to have, and this is speculative at this point because we don't know what's the answer. So why we want to have a phase separation of the proteasome to promote cell death under conditions of nutrient deprivation. So we think that in the context of tissues, we want to save cells from that. If there is, for example, drain wound, we want to save cells from, some cells from, and although perhaps by having some cells that they basically die and release their components for other cells to promote the survival, for example, amino acids or other nutrients. So that can basically allow the other cells to survive. And you can also think about it as a tumor suppressor mechanism, because if we have cells that they have experienced drastic conditions, for example, nutrient deprivation, we can have perhaps permanent epigenetic or genetic aberrations that can promote cancer. So in this sense, it can be perceived as possible tumor suppressor mechanism. And with this, I want to thank all the students, Maxime Uriart, who has mainly, and Nadine, they have been involved in doing the work, and all the colleagues that I collaborate with, and thank you for your attention.
All right. Thank you so much for that talk. I invite anyone who has any questions to feel free to raise their hand and we will unmute them. Okay. Go ahead, Gabriel.
Dr. Affar, great presentation. Just a quick question, maybe confirmatory, because I might have missed something here. But in slide 16, you had all of the different treatments of your cells, right? With MG132, with a bunch of different inhibitors. And I noticed that when you treated your cells that would create foci with MG132, those foci become ablated, like they're not there anymore. Is that true?
With MG132, we will increase their size. So the MG132, we promote we promote them. For example, if we take a time point after nutrient deprivation, for example, 2 hours time point where we don't have that much foci, and if we have in addition MG132, that will basically promote them. And we think that this is because we promote ubiquitination, and ubiquitination, we prevent actually the de-ubiquitination, and having this increase of ubiquitination will basically promote the formation of the foci.
So that's interesting, because to me it maybe seems like K48 linkages are kind of the scaffold for creating more and more foci to aggregate together. But then it's interesting that normally ubiquitinated proteins in the nucleus, like CG and like you mentioned before, those ones become rescued even if those foci get created. So can you comment on, like, I'm still confused as to whether these proteasomes are actually functional, because it seems paradoxical that even if you can degrade K48 linked proteins, that these foci still form in these cells. I don't know. There's something I'm missing here that doesn't.
Yeah, well, thank you. That's a good question. We are still investigating the impact of the ubiquitination in this process. We know that ubiquitination is required because if we block the proteasome, we increase them, and if we put the E1 inhibitor, we prevent them. And we know that perhaps one way to think about it is to say that there are a lot of ubiquitinated proteins that, for example, in conditions of nutrient deprivation, that they will be accumulated and that will promote their formation. So whether now the proteasome is active in this foci, we believe that it is. To which extent it's more active than the soluble one, we don't know. But hopefully we can do some reconstitution experiment in vitro of the proteasome, and we can try to test the activity of the proteasome in this context. But we know that the proteasome is active because, for example, also when we manipulate the half-life of proteins, nuclear proteins, in conditions where we have almost essentially all the proteasome in foci, we know that we can impact the stability of proteins. So we know that it's active.
Very, very nice. And Kathy, if I could, just one extra question. So I know, and you mentioned it too, that when you restore foci formation, either by reintroducing salt or kind of inducing them in that sense, they go back to very specific locations within the cell. I mean shown through the IF images. Can you comment a bit on some of the, I don't know, the macromolecular environment that it goes into? Like, is these like laminin associated domain regions where there's a bunch of heterochromatin? Like, what's going on there?
Yeah, well, these are good questions. We know that for example, when we try with osmotic stress, and some other colleagues found that as well, that there is some attachment of this proteasome foci to the nuclear envelope. But in the nucleus, for example, in many cases, for us, under conditions of nutrient deprivation, we see them in the achromatic regions. We don't see them in heterochromatin. So we don't know yet. Because of course, when you talk about these foci, and also taking into account the resolution of the microscopes, when we manipulate them dynamically, and we say that with what we're using as a microscope, unless we, and hopefully in the future we will be doing some super resolution microscopy, we can say that they completely disappear. For example, if we do a hypotonic buffer, we see them disappearing and reappearing again in the same place, but we cannot say that they completely disappear. So I think we have still to do, to understand, to define marker dynamics.
Okay. Thank you very much. Appreciate that.
Okay. I see we have Dr. Charette. Go ahead.
It's a fantastic talk. Thank you very much. So my question was much more of a technical one. We are kind of newcomers to the field of intrinsically disordered regions, and I'm wondering if when you use them in the cell context, not in the protein purification context, did you look to see if your truncations where you were removing the IDRs, if those proteins were destabilized? And I'm asking that because we've run into this problem with one of our IDRs where when we truncate it basically in the cell strain, in yeast in our case, the protein, like there's almost nothing left. And so I'm wondering if that's something that's been looked at, because it could obviously influence the interpretation of your results.
Yeah, well, thank you. That's a good point. Ironically, I will say, we're doing this phase separation and we didn't touch yet the disorganized regions. We started with the organized domains because we had a good rationale for starting with them, taking into account that they will provide the multivalency to promote this liquid phase separation. But we know that RAB23B and likely other proteins will be involved as well. They have these organized regions. So we have the mutants, but we didn't check them yet. But that's a good point.
Thank you.
All right. And our next question comes from Claire. Oh. Claire, if you have a question, feel free to unmute yourself.
Sure. It wasn't letting me unmute for a sec. That was a great presentation. Do you have any idea or plans to look into what other proteins are in these phase separated foci?
Yeah, that's a good question, Claire. Actually, as we speak, the students in the lab are doing generating some construct to do some proximity labeling to do the proteomics of these foci. We had some problems isolating them because they are extremely dynamic, and each time we interfere with detergent, with hypotonic buffer, everything that we do in general to do the lysis, we see them dissipating. But yet we will still do some purification and see what we can get with classical tandem affinity purification based proteomics. We will do also some turbo ID labeling and see, but that will be that will be fantastic to know what's happened, what we have in this foci. We have one question in the chat by Kerry. Can you say a bit about RAD23B in cancer or cell survival? Yeah, that's a good question for cancer. RAD23B is involved in DNA repair as well, in nucleotide excision repair. And we can, we like to think about it as tumor suppressor. So we would expect that it will be down-regulated or mutated. There are some mutations in cancer. We didn't analyze them carefully yet. Also, the expression of RAD23B, I think something that we can check more carefully. But in terms of patient survival, these are things that we didn't do. In terms of cell survival, yes, we know that when we prevent RAD23B, when we deplete RAD23B, we promote cell survival. So it seems to be a pro-apoptotic factors that will promote cell death in response to nutrient deprivation. So this is why we think about it as a tumor suppressor mechanism.
All right, I don't see any other questions in the chat or from the audience. So with that, I'd just like to thank our two speakers, Louis and Dr. Affar, one more time. And thank you for all the participants who came out today. And we look forward to seeing you all in our June seminar. Thank you and have a great day.
Thank you. Thank you for having me.
Thank you.
Thanks for the talk. It was great.
Thank you, everyone. We will be closing the Zoom call now.